Imagine a massive manufacturing plant churning out like millions of highly complex products every single day.
Right, a totally massive scale.
Yeah, and it operates flawlessly 24/7 for your entire lifetime. But if you walk into this factory, there's no master clock on the wall.
No manager in an office anywhere?
Exactly. There's no manager orchestrating the shift changes. The entire massive assembly line it coordinates itself locally perfectly without a central brain telling it what to do.
Which, when you think about it, is just completely wild.
It really is. So, welcome to this deep dive. Today, we are exploring this genuinely breathtaking paper from the journal Cell. It's titled The Temporal Architecture of the Seminiferous Epithelial Cycle Revealed by Spatial Transcriptomics.
Quite a mouthful, I know.
Yeah, definitely a mouthful. But our mission today is to decode this profound biological mystery. We want to figure out how the mammalian body uses physical space to keep perfect time.
And, it is honestly one of the most elegant systems in all of biology. We're talking specifically about the mammalian testes here, which acts as this continuous, never-ending assembly line for producing sperm cells.
Right.
And understanding how this localized timekeeping works, it goes way, way beyond just reproductive biology. It answers these massive fundamental questions about biological robustness,
Like how tissues just keep going without breaking down.
Exactly. It explains how tissues manage to regenerate and how stem cell niches can function without a single error for decades. It forces us to just totally rethink how tissues organize themselves on a structural level.
Okay, so to understand the clock, we first have to understand the map. So let's establish the anatomy we're dealing with here.
Sure. Let's lay the groundwork.
We're looking at spermatogenesis, which is the entire journey of a baseline stem cell transforming into a fully mature, swimming sperm cell. In mice, that entire journey takes about 35 days.
Right, 35 days from start to finish.
But the factory floor itself operates on this repeating schedule, the seminiferous epithelial cycle, that takes exactly 8.6 days to complete.
Yeah, and that 8.6-day cycle is sliced up into 12 highly distinct stages.
Wait, so the whole journey is 35 days, but the cycle is only 8.6.
Yeah, it's like, well, think of overlapping shifts. And this is where the geometry becomes crucial. Picture a tiny microscopic tube called the seminiferous tubule.
Okay, I'm picturing a tiny tube.
If we take a cross-section of this tube, we find the stem cells, the spermatogonia, sitting on the very outer edge.
Yeah.
That boundary is called the basement membrane.
The outside wall of the tube.
Exactly. And as these cells develop, they physically move. They divide and differentiate, migrating radially inward toward the hollow center, the lumen of the tube.
So their physical location essentially equals their developmental time.
Precisely.
Like the closer they are to the center, the further along in that 35-day journey they are. But the factory isn't just one single cross-section.
No, not at all. Down the entire length of this tubule, there's something called a spermatogenic wave.
A wave?
Yeah, the 12 distinct stages of development are organized sequentially down the length of the tube, almost like a wave rolling through a sports stadium.
Oh, wow. Okay, so that staggering must be vital, because if every cell in the tube matured at the exact same moment, the body would just release sperm in massive sporadic bursts with long gaps in between.
Which would be terribly inefficient. By organizing the 12 stages sequentially down the length of the tubule, the system ensures a continuous, steady release of mature cells into the center.
So historically, if researchers wanted to understand the genetics driving a complex tissue like this, they'd use single-cell RNA sequencing, right?
Yeah.
shRNA-seq.
They would, yeah. That was the gold standard.
But that tool has a pretty catastrophic flaw when you're trying to study spatial architecture, doesn't it?
Oh, it really does. Single-cell RNA sequencing requires you to completely dissolve the tissue.
Yikes.
Yeah, you essentially put the tissue in a chemical blender, break apart all the individual cells, and then read their genetic material.
So you get a great inventory of what types of cells are present, but you totally destroy their physical context.
Exactly. You lose the map.
It sounds like tearing up a map of a city, throwing all the pieces in a pile, and then trying to count how many coffee shops there are. I mean, you know the coffee shops exist, but you have no idea what street they're on or what they're next to.
That is the perfect way to visualize the limitation, honestly. So to solve this, the research team used a revolutionary technology called CFSH plus NAIR.
Okay, let's unpack this. What exactly is CFSH plus that?
It stands for sequential fluorescence in situ hybridization. It's a form of single-molecule spatial transcriptomics.
Okay, that's a lot of big words.
Basically, instead of putting the tissue in a blender, they leave the tubule perfectly intact on a glass slide. Then they use highly specific fluorescent probes that bind to the RNA right where it sits inside the cell.
I want to make sure I'm grasping the scale of this because the numbers in the paper are just staggering.
They really are.
The probes are essentially lighting up the genetic blueprints as they're being read, and they profiled 2,653 different genes across more than 860,000 adult mouse testis cells.
Yep, 867,062 to be exact.
Unbelievable. And they identified 26 major cell types, all while preserving the exact microscopic GPS coordinates of every single cell.
It gives you a pristine, high-definition map. Because they have that map, they could prove that it takes exactly 4 complete 8.6D cycles for a single germ cell to transition from the outer wall to the mature inner lumen.
I love visualizing this like a sushi conveyor belt.
Oh, that's a great analogy.
Right. If you understand how the kitchen's conveyor belt is laid out, you can tell exactly what time it is in the chef's production cycle just by looking at what stage of sushi preparation is passing by on a specific plate in front of you.
Exactly. By looking at a perfectly intact cross-section of the tubule with CTVH plus mass of AMP, researchers could reconstruct the entire temporal cycle.
Okay, let's do a quick recap here for everyone listening. The core idea so far is that physical space in this tissue literally maps to time. We've got an 8.6-day cycle, 12 stages, moving from the outside wall to the inside center.
And CFHH Plus is the tech that let us actually see this map without putting it in a blender.
Exactly. So here's a question for you listening right now. Think about your own understanding of biology. Did you ever consider that physical location could be a literal substitute for a clock? It's pretty wild to think about.
It completely shifts our perspective on biology.
Yeah.
But, solving that only opens the door to the next, much larger mystery.
Which is?
Well, if space equals time in this factory, who or what is actually keeping the time?
Oh, right. Because when we look at those 26 cell types identified in the map, the vast majority are germ cells, right? The ones actively becoming sperm.
Yes, the vast majority.
But there are also somatic cells, the non-sperm support cells. And the spatial data revealed something that completely shifts the paradigm. The Sertoli cells are the only somatic cells in the entire testis that synchronize precisely with the 12-stage cycle of the germ cells.
Sertoli cells are architecturally fascinating. They're these massive, tall, column-like cells that stretch all the way from the basement membrane on the outside, completely spanning the distance to the lumen in the center.
Like massive pillars.
Exactly.
Yeah.
They physically cradle the developing germ cells. And what SeQI's Plus eliminated was that their internal gene expression is perfectly oscillating to match the 12 stages of the germ cells surrounding them.
And this synchronization isn't just like structural support. They are operating an intricate mechanism that the researchers call the blood-testis barrier treadmill.
The biology here is wild, but it makes complete sense when you think about the immune system.
Right, because developing sperm cells are haploid. They only have half a set of chromosomes.
Which means the body's immune system does not recognize them as self. If white blood cells detect these haploid cells, they'll view them as foreign invaders and just attack them.
Which would be bad.
Very bad. To prevent an autoimmune disaster, the Sertoli cells have to create an impenetrable, airtight seal near the outer wall. This is the blood-testis barrier.
But the developing germ cells, specifically the preleptotin spermatocytes, have to physically move from the outside of this barrier to the inside.
Hold on. Preleptotin spermatocyte is quite the mouthful.
It is, yeah.
What exactly is that cell doing at this stage that makes it so critical?
Well, a preleptotin spermatocyte is essentially a germ cell that is just beginning the process of meiosis. That's the cell division that will eventually have its chromosome count.
Oh, okay.
So it's right at this developmental threshold where it needs to cross the barrier to continue maturing in a protected environment. So the structural paradox is, how do you move a solid object through an impenetrable wall without ever breaking the seal? It sounds exactly like a space station airlock. You can't just open the door to the vacuum of space. You have to seal the room behind the astronaut before you open the door in front of them.
Yes. And the spatial data shows us they execute this airlock maneuver with astonishing genetic precision. During stages 2 through 7th of the 12-stage cycle, the Sertoli cells express something called the scribble complex.
Scribble complex.
Yeah, this genetic network builds and maintains the bottom or basolateral wall of the airlock, keeping it firmly sealed beneath the migrating germ cell.
And then right as the cycle hits stage 7th, the CFHH plus probes show a completely new set of genes turning on, right?
Yep. This is the PAR complex, driven by genes like CDC42, MAGE1, and MAGE3.
Okay, and what do they do?
They activate to build a brand new top wall, the apical junction above the germ cell.
Wow. Once that new ceiling is built and secured, the Sertoli cell uses another specific protein, RGAP17, which acts almost like a microscopic demolition crew. It disassembles the floor beneath the germ cell.
And the cell moves upward, and the barrier never ever leaks.
Wait, hold on. This is brilliant engineering, but are we sure the Sertoli cells are actually in charge here?
What do you mean?
Well, isn't it more likely that they are just passively reacting? Like an automatic sliding door, a germ cell bumps into the barrier, and the Sertoli cell just reacts by opening the airlock.
See, that was the traditional view for decades. The assumption was that the developing germ cells were the master regulators, and the Sertoli cells were just passive scaffolding reacting to physical cues.
But the data says otherwise.
Yeah, if we connect this to the bigger picture, the Seminiferous H plus data revealed something completely different. The perfectly timed activation of these complex genetic networks suggested the Sertoli cells weren't just reacting. They appeared to act as active coordinators, not just a passive structural scaffold. They have an intrinsic program.
Okay, let's pause for another quick recap. The Sertoli cells are basically massive pillars holding the factory together. They run a precise genetic program to move germ cells through a tight airlock called the blood-testis barrier treadmill using the scribble and par complexes.
And they seem to be actively driving the process, not just reacting to it.
Right. So, listener, think about this for a second. If you were designing a factory, would you have the walls physically move the products, or the products move through the walls? Because the Sertoli cell is essentially a moving wall. It's incredible.
It really challenges our basic assumptions about tissue architecture.
Okay, but a correlation in timing doesn't prove who is holding the stopwatch. How did the researchers actually prove the Sertoli cells are the true master clock?
To figure out if the Sertoli cells are the master clock, they had to design experiments that removed the germ cells entirely.
Take away the orchestra.
Exactly. If the Sertoli cells are simply reacting to the germ cells, then removing the germ cells should flatline their activity. The clock should stop. They used two massive, exhaustive models to create germ cell depleted environments.
The first was a chemical ablation model using busulfan. Now, the paper describes busulfan as a DNA alkylating agent. How does an alkylating agent actually discriminate between the cells it kills and the cells it spares?
An alkylating agent essentially damages the DNA structure. When a cell attempts to divide and replicate that damaged DNA, it triggers a self-destruct mechanism.
Oh, I see.
Because the germ cells are continuously rapidly dividing, the busulfan wipes them out completely. But the Sertoli cells in an adult testes are postmitotic. They've completely stopped dividing.
So the drug leaves them structurally intact but completely empty of their germ cell payload.
Exactly.
That is highly targeted. And they didn't just rely on the chemical method, they backed it up with a genetic approach using WWV mice. These mice are born with a congenital mutation in the KIT gene. Why does that specific gene matter?
The KIT receptor is absolutely essential for germ cells to migrate into the developing testes when the mouse is still just an embryo.
So they never even make it there.
Right.
Yeah.
Because of this mutation, the germ cells essentially get lost. They fail to colonize the testes in the first place. So the researchers have two parallel models, one where the germ cells are chemically destroyed and one where they never arrived at all.
Okay, picture looking down a microscope at these completely hollow tubes. There are no sperm cells maturing. The factory floor is completely empty. But when they applied the CPH plus probes, the fluorescent lights still lit up in perfect sequential waves down the walls of the tube.
Astounding.
The Sertoli cells still maintain a cyclic transcriptional profile. They were still cycling through the stages.
It's a remarkable reveal. In both models, the master clock is undeniably intrinsic to the Sertoli cell. Think of a phantom orchestra.
Oh, I love this analogy.
The Sertoli cell is the conductor on the podium. The germ cells are the musicians. You can clear the entire orchestra off the stage, leaving the chairs completely empty, and the conductor is still up there passionately waving the baton, keeping perfect tempo in an empty room.
It changes how you think about biological resilience, a system prioritizing a complex running program even when the primary payload is missing shows an incredible evolutionary failsafe. Let's do another quick recap. We looked at the busulfan chemical model and the WWV genetic model. Both removed the germ cells, but the Sertoli cells kept ticking.
The conductor kept waving the baton.
Exactly. So to you listening, what does it tell you about biological resilience that the system keeps running a program even when the primary payload is missing? Nature doesn't mess around with its backups.
It really doesn't. But, the researchers did notice a slight change in the conductor's rhythm in these empty tubes.
Right. The data revealed what they called reduced amplitude, and gene-specific dephasing. While the intrinsic cycle was absolutely there, it lacked its usual razor-sharp precision.
Right. The highs of gene expression weren't quite as high, the lows weren't as low, and the clear boundaries between the 12 stages became a bit blurry. The rhythm got loose because the conductor wasn't getting any acoustic feedback from the musicians.
The clock ticks on its own, but it gets sloppy without the environment interacting with it, which leads to the deepest mechanism in the paper. If the conductor is keeping time without the orchestra, what is powering the conductor's internal metronome?
What are the actual chemical gears turning this clock inside the Sertoli cell?
Yeah.
Well, the engine of the entire operation is retinoic acid, or RA, which is essentially a highly active derivative of vitamin A.
Okay, retinoic acid.
The spatial transcriptomics illuminated a beautiful three-phase chemical engine inside the Sertoli cells, driven by a metabolic feedback loop.
To build an oscillating clock, you need something that builds up and then breaks down, right? You need a tide.
Exactly. And the precision comes from specific enzymes. You have the builder's enzymes, synthesized by the ALD1A1, ALD1A2, and ALD1A3 genes. These create retinoic acid.
And counteracting them, you have the destroyers.
Right. Enzymes from the CYP26A1, B1, and C1 genes, which degrade retinoic acid.
And it plays out in three distinct phases. Early in the cycle, the builders and destroyers are balanced. Production equals degradation.
But in the middle phase, the builders go into overdrive. Production violently exceeds degradation, creating a massive spike in retinoic acid.
And finally, in the late phase, the destroyers take over, wiping the slate completely clean. This rising and falling chemical tide happens continuously, even in the mice with no germ cells.
Identifying the tide is one thing, but proving it powers the clock requires stopping it. The researchers introduced a highly specific drug called WWIN 18446.
And what does that do?
This compound completely blocks the ELT enzymes from synthesizing retinoic acid. It shuts off the builders.
And the result was dramatic. The clock didn't just get sloppy, it completely froze. The entire Sertoli cycle collapsed and halted right at the mid-cycle phase, around 180 degrees on their circular data map.
The conductor's arms just stop mid-swing.
That freezing proves that retinoic acid is a permissive signal. It is the core fuel of the oscillator. Without the retinoic acid tide, the intrinsic clock cannot progress forward at all.
So retinoic acid is the engine that permits the cycle to run. But we established that in a normal, healthy testes, the clock is much sharper than it is in an empty tube.
Right, the blurry boundaries we mentioned.
If RA is the wrong engine, what provides the tuning?
How does the cycle stay sharp?
The tuning comes from the germ cells themselves. As they grow inside the Sertoli cell's embrace, they send paracrine signals back to the host. They're actively communicating with the wall.
The musician's talking to the conductor.
Exactly. The researchers identified a specific signaling pathway called WNT. That's WNT, emanating from the germ cells.
And to isolate the effect of that specific communication, they used another drug, LGK974, which blocks the secretion of WNT.
And the contrast with the retinoic acid experiment is striking. When they blocked WNT, the clock did not stop. The retinoic acid engine kept turning.
But the cycle began to look exactly like the germ cell depleted mice. The temporal wave became compressed, the gene expression got sloppy, and the distinct stages dephased. So what does this all mean? I have a mnemonic device to help remember this.
So let's hear it.
Think of the word RAW, RAW. The R stands for retinoic acid, which is the engine, the fuel, and the W stands for WNT, which is the steering wheel.
That is a phenomenal way to frame it. You have retinoic acid acting as the permissive engine, the fuel that forces the cycle to exist in turn, and you have WNT signaling acting as the refining steering wheel, the environmental feedback that keeps the cycle perfectly sharp and on track.
Okay, final recap time. We've got the RAW system. Retinoic acid runs the base clock, and WNT refines it.
Permissive versus refining.
Yes. So, listener, think of the systems in your life. Which ones are permissive, like they turn the system on, versus refining, the ones that keep the system accurate? It's a great mental model for basically anything.
When you synthesize all of these findings, you arrive at what the authors call the integrative model of the seminiferous epithelial cycle.
The grand unified theory of the factory.
Exactly. At its baseline, the Sertoli cell possesses an intrinsic, unstoppable biological oscillator powered by the metabolic tide of retinoic acid. It is a metronome ticking away in the dark.
But under normal conditions, the growing germ cells provide localized feedback signals, like WNT.
Yes. This feedback refines and reinforces the underlying rhythm, creating a highly robust, high-amplitude cycle. It's a perfect blend of autonomous timekeeping and environmental tuning.
It makes you think about how we design our own systems, doesn't it? Whether it's supply chains, software, or organizational structures, we almost universally default to a top-down, centralized clock. We want a manager.
We love a master control room.
Right. But biology proves that a decentralized architecture, where the structural support cells contain an autonomous engine refined by local feedback, is infinitely more resilient. If a minor disruption happens on the factory floor, the system doesn't crash waiting for orders from the brain.
No, the local clock just keeps ticking on its base, retinoic acid fuel, until the white signals recalibrate the precision.
This brilliant decentralized architecture is why stem cell niches can function flawlessly for an entire lifetime. It's built for ultimate resilience.
It really is. And, this raises an important question to leave everyone with.
Oh, I like where this is going.
If spatial transcriptomics just revealed that the support cells in the testes are secretly running a master clock, what other boring support cells in your body are doing the same?
Like the ones we just ignore.
Exactly. The cells in your gut, your hair follicles, your brain, are they secretly holding the master clock for your other organs?
We usually look right past the support structures to focus on the flashy dividing cells. But the architecture of the walls might actually be dictating the entire schedule. It is an incredible reminder to keep questioning the hidden architectures of the world around you. Thank you for joining us on this deep dive.